All right, so good afternoon, everyone. I'm John Guatelli, and I want to welcome you to the 2021 Rick Mitchell Memorial Lecture, which is presented today by Dr. Molly OhAinle from the University of Washington, Seattle. First, I'd like to offer some thanks, first of all, to the San Diego CFAR and HIV Institute staff, including Bryna, who you've just heard from, and Bryna Block, Kim Schoonover, and Liz Johnson for setting up this seminar and this series. And you all for coming, and to many of you who have contributed to support this series. And a special thanks goes in that regard to Gail and Skip Stokes, Rick's sister and brother-in-law, for their support, not only of the series, but in general. And that includes their goal of establishing a Richard S. Mitchell Endowed Postdoctoral Fellowship. So I'd just like to say a few words about Rick. Rick was a postdoc in my research group at the time of his untimely death 12 years ago. We lost not only an excellent researcher, but a really great friend. Rick was a great lab mate. He had this wonderful, mild-mannered, modest personality and infectious wit, and he was a very good teacher. He always went out of his way to help his students, and anyone in the lab. So he remains greatly missed. Rick obtained his PhD working with Rick Bushman at the Salk Institute through UCSD. And up here on the top is a seminal paper from his PhD work on retroviral integration sites. This really set a standard for the use of deep sequencing and bioinformatics in this field. And it remains highly relevant today, since integration is really the central feature of HIV persistence in people. In my lab down below, this paper was one of Rick's biggest, most well-referenced. Here, Rick studied one of the HIV accessory proteins named VPU, viral protein U, and how it counteracted a host cell so-called restriction factor, the interferon-inducible protein BST2, also known as tetherin. Rick studied this in great detail. And it's really something that this kind of thing, the host pathogen relationship at the level of cellular proteins, is something we're going to hear a lot about today, I think. So in that regard, we're very happy to remember Rick in this way, and extremely happy to have Dr. Molly OhAinle for today's lecture. Dr. OhAinle obtained her Bachelor's of Science at UC Davis, and then went on to the University of Washington and got her PhD in 2007, studying molecular and cellular biology under the guidance of Drs. Michael Emmerman and Harmeet Malik. She then went to UC Berkeley for a postdoc in the School of Public Health before returning to the University of Washington and working at the Fred Hutchinson Cancer Research Center as a staff scientist. She's now an assistant professor at the University of Washington in the Department of Global Health. So over these years, Dr. OhAinle has gotten off to a great start in her young career, complete with NIH funding, independent funding, and some very exciting papers. And I think she'll show us some fabulously clever techniques to really get into the host pathogen interaction at the cellular level, and to find out what host restriction factors exist and how they work together to affect HIV replication. So with that, Molly, we're very happy to have you, and we're looking forward to your presentation.
Great. Thank you, John, for that very kind introduction, and it really is an honor to be here. I did not know Rick, but I've heard about him from others, and he seems like he was certainly a tremendous scientist and perhaps an even better person, and very well liked. And I'm really, it's an honor for me to be here in his honor giving this seminar today. So as John alluded to, today I'm going to tell you about my work to try to better understand how it is that our cells are able to block infection by viruses such as HIV. And I'll show you a new sort of approach that I developed to do this, and share with you some of our findings from that work. So first and foremost, my interest in virology really stems from this really kind of simple feature of virus replication, which is that viruses are sort of require our host cell machinery to replicate themselves. So they're obligate intracellular parasites. They must get inside our cells, use the machinery that's there along with their own encoded machinery to make more of themselves. And they do this sort of fascinatingly with a relatively limited coding capacity, meaning that they only have so many genes to play with of their own. And so therefore they really do heavily rely on the host cell biology to replicate their own genomes. And so they sit at this precipice also of, while they rely on our host cell machinery, they also must contend with this arsenal of host defenses that aim to actually directly inhibit virus replication. And it's these host defenses that I'm going to talk to you about more today. So this is an example of the HIV-1 sort of representation of the life cycle. And so like all viruses, it must get in the cell to replicate itself. So HIV binds on the host cell surface to the CD4 receptor, enters the cytoplasm, must get into the nucleus where it's going to take its reverse transcribed genome and insert it into the host cell chromosome. And from there be transcribed and translated. These proteins and RNAs then go to the cytoplasm where they're able to then bud out from the cell. And in the HIV field, we often refer to factors that the virus requires for replication as dependency factors because the virus depends on them to replicate. And so these host factors mediate various steps of this virus life cycle: entry, transit, transcription, translation, and leaving the cell, the egress process. And again, my work is really focused on the other end of this cell biology, and that is to understand these family of really cool antiviral directly acting host cell proteins. And we know of a number of examples of these genes, and some of them I will talk to you about today, where these proteins can actually directly impede sort of the ability of the virus to transit through the cell. They can mutate and silence viral genomes. They can directly degrade viral gene products, and they can even tether, which is the interest of Rick Mitchell's work in John Guatelli's lab. They can tether particles to the viral surface. And so one of the more fascinating aspects of the biology of these genes is that in order to successfully infect a cell, the virus must figure out ways of directly either evading or antagonizing these host cell restrictions. And on top of that, if we look at sort of how these host genes evolve in our genome related to our close relatives, the other primates, we find that these restriction factors are some of the most rapidly evolving genes. And what I mean by that is that if you were to take a version of this gene from our genome and compare it to a related primate, closely related primates or more distantly related primates, what you find is that those gene sequences actually can vary quite a bit. And the reason this is important is that we know that viruses have to evade or antagonize these restrictions to infect these cells. And specifically in terms of HIV infection, we know that HIV is actually a virus that originated in primates in Africa from a simian immunodeficiency virus. And one of the aspects of my work and the work that I will continue to do is to try to understand how it is that these viruses were capable of entering the human population and replicating themselves. So this happens both in terms of sort of zoonotic transmission events as well as person-to-person transmission events, which is some of the data I'll show you today. So we think that overall, that the viruses are constantly trying to adapt to the host cell machinery present in cells. And so all of these transmission events, whether it's from primates to humans or from human to human, these transmission events do require virus adaptation. So the overarching sort of theme of the work that I do is that I'd like to understand how it is that host cells restrict viral infection and how it is that the virus is able to overcome these barriers. And in our work, we use sort of two major approaches that I'll tell you about today. The first is a functional genomics approach. So this is a CRISPR screening approach specific for HIV that I developed and I will tell you more about today. And we pair that with more standard sort of molecular virology, where we're gonna sort of take apart and look at various aspects of how the virus life cycle is occurring in the context of these factors. And we're always sort of keeping in mind how the virus and these primate genomes are evolving and what that means in terms of how these infection events are occurring. So today I'll tell you primarily about some work that is really focused on trying to understand a major component of how our cells respond to viral infection, and that's through the type 1 interferon response. So our cells have a number of different mechanisms of sensing virus infection, and when that happens, they produce these sort of danger signals mediated by interferon alpha and beta, or the type 1 interferons. And these molecules then can act back on the cells that are infected as well as surrounding cells to turn on these gene programs of interferon stimulated genes. And so these genes, these ISGs, are sort of well characterized to have the ability to block virus replication. So by turning on these genes, our cells are sort of turning on mechanisms by which they can be protected from infection. And this is particularly important. I wanted to give some context to why, in the context of HIV infection, this is such an important aspect of work to study. And that largely stems from recent work from the Hahn lab, from Beatrice Hahn's lab, looking at the ability of different types of HIV-1 to sort of contend with the type 1 interferon response in HIV-infected individuals. And so what I'm going to show you here in a couple snippets of her data is that interferon resistance, or the ability to sort of contend with these ISGs, is one of the primary properties that's selected for during transmission events and during viral rebound in HIV-infected persons. So that data is shown here. On the y-axis, this is a measure of how much interferon is required to inhibit 50% of sort of virus infection. And so the higher you are on this scale, the more interferon resistant you are. And every dot on this plot is a single virus isolate, isolated from a patient. And in dark gray, these are successful transmission events. So these are viruses isolated during acutely infected individuals in a prospective cohort. And as compared to the sort of viruses isolated from individuals chronically infected with HIV in the light gray, what you can observe is that one of the strongest phenotypes they found was that these viruses that are successfully transmitted have high interferon resistance. So they have the property of being able to survive in the face of a large dose of type 1 interferon. And sort of on top of this, in more recent work from the Hahn lab, they were able to observe that when they take virus isolates from patients that underwent analytical treatment interruption, so they were taken off their antiretroviral drugs, and they take viruses isolated from those patients, they observed that these are some of the most interferon resistant sort of HIV-1 isolates that have been observed. And so these rebound viruses have the highest interferon resistance. And so what I'm going to tell you is that those restriction factors that I introduced you to, many of these are themselves interferon stimulated genes. So their expression gets turned on by type 1 interferon. And taking this all together, this tells us that evasion and antagonism of ISGs is a key and ongoing process in HIV infection and transmission. So while I have an interest in sort of what happened in terms of HIV becoming a human pathogen and its emergence from SIV reservoirs, it's also clearly a very important sort of feature of HIV biology, even in sort of chronically infected HIV-infected individuals, as well as in these transmission events and rebound events. So that's just to give you a little context to why we care so much about interferon. Okay, so taking all that complex biology aside, we wanted to move into a cell culture system in the lab where we could try to model what's happening in terms of this sort of interferon response to infection and what's going on. And so we can do this quite easily in the lab. This is an example of an experiment in THP1 monocytic cells, where we can take cells and either pre-treat them with interferon, shown in the dark gray circles, or we just simply infect these cells without any interferon. And on the y-axis, this is a measure of how much virus is being made in these cells, looking at the P24 capsid sort of released into the supernatant. And so if we look across days of infection, so 2 days post-infection or 5 days post-infection, in the absence of interferon, the virus can spread quite well through the culture. But in the presence of interferon, there's a really robust inhibition of virus replication. And so this gives us sort of a hook to try to understand these gene programs that are leading to inhibition of virus replication. And so we started with a really simple question, which is, we think that these interferon stimulated genes are leading to this reduction of replication. And we'd like to try to identify what are the genes that are actually mediating this effect. And so you can imagine that there might be many genes that are responsible for this inhibition. So a number of genes that are working together. There could be just a few. There could even just be one gene that's primarily responsible for inhibiting virus replication. And so we wanted to try to figure out what they were. So we got to thinking about how does one find what these important genes are in a relatively sort of unbiased way. And so historically, these types of experiments had been done by looking at sort of gene RNA expression profiles and asking about candidates that are sort of highly overexpressed after interferon treatment. And a number of really important findings had been unearthed that way, including some of the factors I'll tell you about today. But we wanted to try to develop sort of a new system to do this in a more comprehensive way. And so we came up with this idea that we refer to as the HIV Crispr Screen, and it has sort of 3 fundamental aspects to it. The first is that we're going to use CRISPR-Cas machinery. So this is a endonuclease from bacteria that can bind to a small RNA called a small guide RNA or sgRNA. And by toggling the little pink part, the 20 base pair sequence in this guide RNA, you can target this endonuclease to a different locus in the genome and allow editing and knockout a function of a gene of interest. And we're going to pair that with some relatively simple but important lentiviral vector engineering that I'll show you, which is going to allow us to actually create HIV CRISPR genomes that can be packaged by HIV-1 in trans. And then third, we're going to do this on a pooled library scale. So one of the benefits of sort of custom DNA synthesis technologies together with high throughput sequencing is that we can ask these questions on a really high throughput and comprehensive way that was not possible before. So to explain to you sort of how these screens work, there's three pieces here. The first is that we can do a really simple experiment, such as the one shown here, where we can introduce the Cas9 and guide RNA machinery to a cell. On the top, I'm showing you an example of a restriction factor knockout. On the bottom are these dependency factor knockouts. And in the middle, in all of our experiments, we always have these non-targeting controls. So they are not expected to be edited in any significant way. And what's important to think about is that if we infect these cultures of cells, so if they have knockouts of different types, and these are experiments many of us have done, including myself, what you expect to see is that in the restriction factor knockout cells, we should see more robust virus replication. And in the cells where we've knocked out a required host factor, such as, for example, the receptor CD4, you expect to see relatively little virus replication. And so this is really important and a really nice way to assay the effect of a gene on virus replication. But what we really wanted to do was to develop a screen where we can do this in a pool of cells where they have sort of a heterogeneous mix of gene knockout. So each cell in the culture will be knocked out for a different gene, but importantly, they're going to be all in one big pool together. And so this raised a really fundamental issue for us, which is that we know the virus will replicate better or worse in different cells in this population. But if we put virus in and simply measure virus coming out, we have no way of sort of disentangling which cells gave rise to more virus and which ones gave rise to less. And so the key to how this screen works is that we're going to actually ask the virus itself to read out the barcodes of which cell it came from. So the replicating HIV virus itself is going to actually tell us which cell it came from, which is going to allow us to know which type of gene knockout had an effect on virus replication. So the way that this works, this is the trick to the screen, is that we re-engineered the lentiviral vector, which is an HIV-based vector that has internal promoters to express the CRISPR-Cas machinery, but we replaced what was missing from the HIV-1 LTR to create this HIV CRISPR construct. And so what's important about this is that in most lentiviral vectors, there are transcription factors that should bind to the LTR, and that region has been deleted. So we simply put that back in to create a wild type or replication competent long-terminal repeat region. And this means that when we introduce this into cells, not only will we express the CRISPR-Cas machinery, but we'll also express a long genomic HIV CRISPR RNA that has, importantly, the 20 base pair sgRNA sort of barcode telling us about the gene knockout in that cell, but also has the Psi or packaging signal of HIV, allowing this genome to be packaged in trans by HIV-1 proteins. And so an example of this is shown on the left, where we're going to create a cell population where each cell is going to have an integrated copy of our HIV CRISPR construct. We will wait a couple weeks so that the CRISPR-Cas machinery can be expressed and gene knockout will occur. And then when we come back and infect these cells, what we're going to see is that there will be a packaged version of this HIV CRISPR genome that can leave the cell with the virus. So in addition to its own genome, this HIV-1 virus will also find and package this HIV CRISPR genomic RNA. And so coming back to this example of the different types of knockout cells, this is important because in addition to having more replication in the restriction factor knockout cells, we will also have an increased representation of the restriction factor targeting guide sequence in that virus population as well. And then conversely, we'll see relatively little representation of guides that target required host cell factors. So it becomes a really quite simple experiment. It's a counting exercise of looking at barcodes, 20 base pair sequences, and whether or not they're enriched or depleted relative to one another. So these enriched barcodes, these enriched sequences are restriction factor candidates, and our dependency factor candidates are the ones that are depleted out of the virus population. So how we do these experiments is we create libraries of guide RNAs, assemble them into our HIV CRISPR backbone, introduce those at a low MOI into our target cells. The data I'll show you today is all in THP1 cells. And this low MOI allows us to select a population where after 2 weeks, we know that each cell in that culture will have a single gene knockout And also each cell in that culture will contain an integrated copy of its this HIV CRISPR construct. So this 20 base pair sequence will be present in the genome. We will then pre-treat with interferon, infect these cells, wait a few days, and then we just simply collect the viral supernatants and the cell pellets. And this allows us to look at how the enrichment or depletion is occurring in the virus population and compare that to what's present in the cells. And the cell pellet is important for us as sort of a representation of the library, so we know sort of what we expect to see from the experiment. So we just sequence these in an Illumina sequencer and then look at the data. So since we were interested in interferon-specific genes, we created our own sort of custom library to look at the role these genes might play in HIV infection. And so we went to existing data generated by other groups, where in HIV target cells, they had treated with interferon and asked which gene, which RNAs are going up in interferon treatment. And through this, we defined a set of about 1,900 interferon-stimulated genes, and then pulled existing guides from other libraries to create a library that has 8 guides targeting every gene together with the non-targeting controls. And again, we just take these 15,000 or so sequences that are synthesized on a chip and insert this into our backbone. And we call this the PICA HIV library, or the packageable ISG knockout assembly. So the data from the screen looks like this. This is an example of one of these screens where we treated with interferon and infected with HIV-1 LAI strain, which is a commonly used sort of lab-adapted HIV-1. And what you're looking at here, each point on this plot is a single 20 base pair sequence from our library. And if you're higher up on this y-axis, we're looking at the enrichment of that sequence in the virus relative to the genomic DNA. And so what you can see is those points that are well above this line are enriched in our viral supernatant, and those below are depleted. And nicely, if I array those non-targeting controls shown in white, you can see those hover right around 0, suggesting that they aren't being significantly enriched or depleted in the virus at all, which is exactly what we would expect. And so these restriction factor candidate genes that we'd like to identify should be present sort of above 0, and dependency factor targeting guides would be present below. And I'm not really going to talk much more about this today due to time, but we can use our screens to identify required host factors in addition to looking at restriction factors. So we then take data from these screens where we can integrate information across all 8 guides for each gene in the library, and use a statistical package developed by another group at MIT to analyze our output of our data and create gene scores for each gene in the screen. And so when we do that, we get data that looks like this in this waterfall plot, where you can see there are a number of genes that are scoring quite highly. So the higher we are on this plot, the more significantly we're scoring to be a restriction factor for HIV. You can see that there's a really large flat line here, suggesting that the majority of knockouts in our population actually had really no effect on virus replication. And these dependency factor genes are here on this bottom part of the curve. And again, I'm not going to show you this, but the very bottom point here is CD4, which is a required gene for HIV infection, and is really encouraging that the screen is functioning sort of as designed. And so I'm just going to zoom in now and sort of look at this top 15 or 20 points on the curve. And so the first thing that jumped out from our data that was really encouraging is that the strongest hits in our screen are 4 components of the type 1 interferon response pathway. So I'm representing that here, where interferon alpha binds to the interferon alpha receptor, and through a number of other mediators, including the STATs and IRF9, transits into the nucleus and drives this interferon stimulated gene program. And so what's important is that if you knock out any component of this pathway, you lose the ability of the cells to respond to interferon, and the virus is actually able to replicate really well in those cells. And so it makes sense that the top 4 genes in magenta are these components of the type 1 interferon pathway. So again, this was really encouraging that the screen was functioning as designed. Then we got to work, getting excited to see what else we might find. And so the first thing I did is I just simply overlaid data for which genes are significantly induced by interferon in the THP1 cells, using that data from RNA-seq and microarray data that we had. And those points now are just blown up here. And if we take a closer look beyond the interferon pathway genes, we wanted to look at what else we were seeing on the screen. And so the first thing that jumped out to us is that there are actually multiple interferon stimulated genes here that are contributing to the effect of interferon. And we know that because if there was a single gene, we would expect it to have sort of fallen out together with the type 1 interferon pathway genes. But instead, what we saw was sort of this array of other factors that are scoring highly together. And so what we found here was that these genes included genes such as MXB, IFIDM1, tetherin, and TRIM5. And so I'm going to tell you a little bit about these. So MXB was actually discovered as an interferon-stimulated gene important for HIV inhibition in THP1 cells. And so it served as a bit of a control and was really exciting that we were able to find this in our screen as well. And so this is a gene a protein that's able to block nuclear import of HIV. We also see IFIDM1, which has been implicated to perhaps be able to block viral protein production. And this gene tetherin, which again was an interest of Rick and John's, and I will come back to here in a minute. But tetherin is able to actually tether, as it sounds, viral particles to the cell surface. And then sort of last, but certainly not least, what we thought was one of the more interesting genes we found in the screen is TRIM5, which is known, a really well-known HIV restriction factor that can block the ability of the virus to uncoat in cells. And the reason this was so interesting to us is that it had been discovered as a restriction factor in the rhesus macaque genome. And so rhesus macaques have a version of TRIM5 that is one of the most potent HIV sort of restrictors that we know of. But many labs had assayed the activity of human TRIM5 and really had kind of decided that it didn't seem to be really doing much of anything. So it was decided that this was not an HIV restriction factor. So this was a little surprising to us that we were now pulling this out of our screen. And so I'm going to show you just one example of what we do with the screen data, which is run back to the lab and set up validation experiments to make sure that these hits we're seeing in the screen are in fact real. And so to do that, we just take individual guides from the screen and we introduce them into cells to create these pooled populations that are knocked out for either controls or for our genes of interest. And then we treat with interferon, infect, and assay for replication. So an example of that here, this is a non-targeting control cell line where when we infect, we get about 50% of the culture, 50% of the cells infected. If we pre-treat with interferon, we see a robust decrease in replication. So in most experiments, this is about a 7-fold inhibition. And so what we saw when we look at the TRIM5 knockout, for example, I just replotted this data so that now you're looking at fold inhibition. So here's that 7-fold inhibition in the non-targeting control cells. If we hit a positive control like STAT2, we completely rescue infection in these cells such that they no longer have any interferon inhibition whatsoever. And what we see with TRIM5, and I'm not going to show you the data, but for the other hits in the screen as well, is that we see this really nice sort of partial rescue. So none of the genes that we identified when knocked out on their own are sufficient to rescue to the level of the interferon pathway genes, but all of them have this sort of phenotype of being about a 2-fold rescue of infection, telling us that the screen is in fact working as designed. Okay, so TRIM5 is an example of a sort of structural model of what we think it's doing. And so this purple lattice that you're seeing is sort of cages around the green HIV core, and it's thought to lead to sort of aberrant uncoating of HIV. So the core of the virus is not able to get to the nucleus, reverse transcribe, and continue its replication. And so what our data suggested, and this has now been sort of corroborated with other labs' data, is that we do think human TRIM5 is a functional restriction factor in human cells. This was really also encouraging because there are some data suggesting there are polymorphisms in humans in human TRIM5 that have been associated with, for example, the ability to become infected, the acquisition of infection. And so it was a mystery for a while why that would be the case if the gene was not doing anything, but we and others now think that human TRIM5 is in fact a highly capable restriction factor, just not as potent as the rhesus macaque version. Okay, so this all left us in a place where we had now created this new method of screening, and it was very clear that it works, and that was very exciting for us. We were able to show that there is a small panel of interferon-stimulated genes in THP1s that are able to inhibit this HIV-1 LAA virus. So this includes MXB, IFIDM1, and tetherin, but also includes human TRIM5, which was sort of a new finding for us and for others. And so together they're mediating this potent 7-fold inhibition of replication. And so what I want to sort of move on to now is just to tell you about how once we created this screen, we started thinking of instantly, what are the other ways that we can use this to get at some sort of fundamental questions? And so by pairing CRISPR-Cas biology with this vector engineering, what we created was a really high throughput assay. And so I like to tell people that these screens can be done by sort of in one flask or sometimes even in one well and by one person. And importantly, the assay, because of the way it's set up, it actually will tell us about the function of genes important in the entire HIV-1 life cycle. And so this is in contrast to other types of screens that were done with reporter viruses, where frequently you're reading out sort of a fluorescent reporter being produced by the virus. And so we're able to see things that affect everything from entry all the way through exit from the cell. We also like that this assay works pretty well in what are close to true HIV target cells, so we can use immune cell line, such as THP1s or other T cell lines we started working with. And then in the next part of the talk, as I'm going to tell you, is that this screen is actually highly portable, so we can use different cells, but we can also use, fundamentally, we thought, any HIV-1 virus. So we're not relying on any type of reporter. So as long as this virus can package our HIV CRISPR vector, we should be able to use this screen against any virus that we'd like. So that was sort of one of the first things we did, is that I just went down the hall here at Fred Hutch to Julie Overbaugh's lab and asked them for a different HIV-1 strain that we could test in our system. So the data I showed you so far, again, was with this HIV-1 LAI strain. It's sort of a really well-studied lab strain, isolated from chronic infection. It's a clade B virus. But we know that HIV sequences, there's a tremendous amount of diversity. And so we wanted to ask, if we use a different virus, this is going to be a clade A isolated from a different time in infection. We want to just simply ask, are other HIV viruses inhibited by the same factors that we just uncovered, or is it possible that they might have sort of different restrictions? And that data now is shown here. And this is one of my sort of favorite things to do with these screens, are these sort of comparisons. So on the x-axis, this is the gene scores against that original virus, the LAI strain, and on the y-axis is the Q23 virus. We see that both viruses, the interferon pathway genes are the highest hits, which is what we would expect. But as we start to really zoom in on what else is present in our data, some interesting things pop out. So again, I'm going to highlight what we determined to be interferon stimulated genes in these cells. And we see that genes like IFNM1, TRIM5, and MXB are sort of scoring relatively highly for both viruses, suggesting that they're in fact restriction factors functioning to block infection by both of these viral strains. But what's really interesting is that there are some differences in this data. And so, for example, on the left, this point IFI16 is scoring really highly against this Q23 strain of HIV-1, but it scores quite low against the LAI strain. And conversely, our friend Tetherin here, that I'm going to tell you a bit more about. But first, the IFI16, after this paper was subsequently shown by Frank Kirchhoff's group, that this is an HIV restriction factor that is very strain dependent. And so the fact that we're seeing the scoring highly against this virus, but not against another virus, actually makes a lot of sense in terms of what is being found by other folks as well. But I wanted to do a quick left turn. John sort of alluded to this paper in the introduction, but I had looked at this paper earlier and wanted to show some of Rick's data that illustrates quite nicely one of the points of this comparative screening that we're doing. So in this paper in John's lab, Rick was able to show that a wild type virus can infect these cultures quite well, but if you delete the viral accessory protein VPU, you lose the ability for virus to sort of get out of these cells. And that's also shown here in this flow plot, where if you stain for Tetherin here on the y-axis, you see really nice staining. If you introduce the HIV-1 VPU protein, the staining drops down quite significantly. So VPU is able to effectively take Tetherin off the cell surface and allow for virus replication to occur. And so this connects really well to the work that we were doing, because what we now know is that the VPU genes encoded by these two viruses are actually quite different. It seems to be a general property that VPUs from primary isolates, such as this Q23 strain, are significantly better Tetherin antagonists. So this ability to bind and bring Tetherin off the surface is something that is much more readily achieved by HIV isolates taken directly from patients, whereas if you adapt a virus in lab, it seems to lose that ability. And so that's why we're seeing that Tetherin can restrict LAI, because it doesn't have a very good VPU, but the primary isolates that we have are not restricted by Tetherin at all. So far what I've shown you is that we have this way to identify these restrictions. We identified a small set of ISGs like TRIM5, in this sort of more recent data, we were able to see that these restrictions can be quite virus specific. We now know that not only does the screening work, but it seems to work for any replicating HIV virus. So I just showed you two here, but I'm going to show you some more data suggesting that it can work against a number of different viral strains. And so this got us thinking again of kind of what we wanted to do with this. And one of the things that I've been spending a lot of time thinking about is ways that we can tweak the HIV and sort of introduce mutations that will change this balance so that perhaps it's less well able to use some host cell machinery or otherwise, and tipping the balance in the favor of sort of host defenses. So by making these simple mutations, can we make poorly adapted viruses that are going to reveal to us what restrictions are present inside of cells? And so let me walk you through one sort of story related to that. So I've become really interested in looking at the early steps of HIV replication. So when HIV gets into the cell, it deposits its core into the cytoplasm. And here again is a structural model of what this looks like. And so one of the really cool aspects of this is that a single HIV protein, the HIV capsid or P24 protein, comes together to form these structures of hexamers and pentamers that form this conical shape of HIV in the cytoplasm. So it's this one single protein that's mediating transit through the cytoplasm, getting to the nucleus. We know also important for reverse transcription and integration into the genome. And so one aspect of this biology is that the virus binds to some host cell proteins to sort of mediate its ability to infect cells. This includes proteins such as cyclophilin A and CPSF6. And I'm not going to talk more about sort of the host cell functions of these, but one hypothesis about why the virus binds to these in cells that's been sort of posited is that the idea that perhaps the virus binds to these host cell proteins to protect itself from restriction by something present in the cytoplasm of cells. And so we were in a really good spot to try to ask if that hypothesis might be correct. And that's because this is a representation of the HIV capsid monomer. And we know of some mutations you can make in capsid. So by changing a single amino acid, for example, from asparagine to aspartate at position 74, we can abrogate the binding of these capsids to the CPSF6 protein. And conversely, by making a single mutation at this site in the capsid from proline to alanine, you can abrogate binding of this capsid to cyclophilin A. So we could knock out the ability of the virus to bind to these host cell proteins by making these mutations. And so by doing that, we can then ask the question, if these mutant viruses become sensitive to some sort of cellular restriction, so are they being shielded from this restriction by binding to these host cell proteins? So we took these viruses into the lab. Here on the left, this is the wild type virus, and then in pink and in blue, we're looking at the inhibition of these P90A or N74D mutant viruses. And so one thing you'll notice is that these viruses actually become hypersensitive to the effects of interferon. So the effects of these ISGs on these mutant viruses is even greater than what we see with the wild type. And so we wanted to use our screen now to ask, can we understand now what is restricting these two viruses? So that data shown here on the x-axis again is the wild type LAI strain, and on the y-axis is this P90A mutation that abrogates binding to cyclophilin A. And so I've sort of drawn this line in here to draw your attention to where we expect points to fall that are shared across both viruses, and then points that fall off the line are those genes that are sort of differentially restricting the two virus strains. So again, we see genes like IFID-M1 and tetherin that are affecting both of these viruses. But what we were interested to see is what is changing. And so one thing that's interesting, and this has been shown by other groups as well, is that when you make this mutation in capsid, you actually lose sensitivity to this MXB restriction. So we see this point fall off the line, suggesting that the P90A mutation makes this virus less sensitive to MXB. But what we also saw was that TRIM5 jumps up off the line, suggesting that this P90A virus is actually becoming hypersensitive to the effects of TRIM5. And so this is data that's been shown also by other groups now, looking at replication of these viruses in primary cells as well, that when you make this mutation, these viruses become sensitive to human TRIM5 restriction. So similarly, we did a similar experiment with the wild type versus the N74D mutant. And again, we see that there are some things that are shared across both viruses. In this case, we do not see a difference in TRIM5 sensitivity. But again, we see a loss of MXB restriction, but gain of restriction by an entirely new gene. And so this gene is TRIM34. And this was exciting for us because it was a completely unknown restriction in human cells, and is now sort of the current focus of much of the work going on in our group, some of which I'll show you here today. Okay, so TRIM34, one of the reasons it was so exciting for us is that it's a close paralog of human TRIM5. So it actually sits on the human chromosome right next door to TRIM5. and even has almost 60% amino acid identity with human TRIM5. And this makes a lot of sense to us because we went looking for potentially new capsid targeting restriction factors, and what we found was a protein that's closely related to a really well-known capsid targeting restriction factor. And so coming back to this model of how we think TRIM5 forms these lattices around the HIV core, what we're focused on now is trying to understand if TRIM34 is restricting through a similar mechanism that we know TRIM5 to restrict. And something sort of arguing in favor of that hypothesis is that if you look at the overall sort of structure of these genes in terms of which domains are present and sort of generally what they look like, there's a lot of homology between TRIM5 alpha and TRIM34. So TRIM34 has all of the sort of necessary components that we think are required for the ability to form these multimers that then form these lattices around the capsid. And it also has an intact Crispr domain, which is the region of TRIM5 known to interact with and bind to HIV capsids. So it has sort of all the basics that it would need to function in a really similar way. I'm not going to show you the raw data, but when we overexpress TRIM34, we can see a block at reverse transcription, which is exactly the same phenotype you see when you overexpress TRIM5. So it does sort of support this idea that it might be functioning in a really similar way. This led us down to sort of even more interesting data that was related to the fact that there were some older papers that had cloned these genes to understand sort of what they might be doing, and actually showing that TRIM5 can interact with TRIM34. And this was shown by immunoprecipitation and these two hybrid data, but that maybe that not only can these proteins multimerize with themselves, but there's a possibility they're actually multimerizing with one another to mediate their effects. And so I'm going to just show you one experiment with some sort of genetic data that sort of highlights this possibility that TRIM5 and TRIM34, at least in some cases, are working together to restrict infection. So I did some experiments in activated CD4 positive T cells, but I'm going to show you data, some really nice data from a former graduate student in Jeremy Luban's lab, where he has the ability to knock down either TRIM5 or TRIM34 using stable hairpins in these monocyte-derived dendritic cells. And so that experiment shown here, in the control knockdown cells, if you infect over a range of sort of increasing virus titer, with the wild type virus, you can see really nice replication. So many cells in the culture get infected. But when you make this N74D mutation, you can see that there is about a log less virus replication in those cells. And so what's really nice is that when Cusick knocked down, stably knocked down expression of TRIM34 with these hairpins, he saw that he could rescue almost up to the level of wild type, the amount of infection of this N74D mutant. So this suggests that TRIM34 in these cells is mediating a potent block to infection by this virus. And this is where it got sort of even more interesting for us, is that when Cusick tried his TRIM5 hairpin, he could see a very similar, and in fact, maybe even slightly higher rescue of infection in these cells. And he even has the ability, again, to do this sort of double knockdown, where he sees a really robust rescue of replication. So what this genetic data suggests is that both TRIM5 and TRIM34 are required for restriction of this virus. And so if you knock out one or knock down one or the other, you lose that restriction. And so again, it sort of suggests they might be working together in a single complex to restrict virus infection. Another avenue we went at to try to answer this question was just looking at, if you put these capsids in an infection in the cytoplasm of cells, do we see sort of localization of TRIM5 and TRIM34 to the same place? And the answer is yes. So working with Ed Campbell's lab, we were able to show that we see TRIM5 and TRIM34 together at a higher frequency with these N74D capsids than we do with the wild type capsid, suggesting again that they might be working together to restrict the virus. So what we think we've unearthed through this sort of using these viruses that are poorly adapted to replicate in human cells is this ability of two TRIMs, TRIM5 and TRIM34, to work together to inhibit virus replication. And so a lot of the work now that I'm not going to talk about today, that's really sort of just beginning, is trying to better understand how it is that these two TRIMs work together to restrict infection and other related viruses. So what I showed you is that we developed a method to comprehensively identify key restriction factors through this approach that we call HIV Crispr Screening. We were able to identify a small panel of ISGs, including MXB, Tether, and TRIM5 that are able to inhibit, so sometimes in a very strain-specific way. We identified TRIM34 as a novel restriction factor that requires TRIM5, and we're sort of avidly working at trying to understand the mechanism by which that might be happening. So what I want to come back to is this idea that the virus kind of sits in this very special place where it's able to use host cell machinery, but also must evade host defenses. And what we think is going on is the virus is sort of continuously having to adapt to pressure from host cell defenses and sort of sit in this really sort of defined space, evolutionary space, where it's able to achieve its goals. And so this is work that we're now sort of going down the road of trying to understand better how it is that SIV has adapted to become a human pathogen. So these poorly adapted SIV viruses, we'd like to understand how it is that they're restricted in human cells. And sort of alluded to with Beatrice Hahn's work, we'd like to better understand how it is that on transmission at these various sort of transmission barriers, we're selecting for viruses that have a very particular phenotype in terms of interferon resistance. So how is it the virus is continuing to adapt as it replicates in the human population? So I'm just going to end by talking about, who's doing some of this work and where we're going with it. And so Joy Twentyman is a graduate student here at UW. And so she's been looking at sort of the role that TRIM34 together with TRIM5 may have played in adaptation of SIV to humans. And so looking at how these genes restrict the related primate lentiviruses and how they might be contributing to these species barriers. Daryl Humes was a staff scientist in Julie Overbaugh's lab and was able to actually get our screening approach to work in primary cells, so in activated CD4 positive T cells. And there's now a graduate student in Julie's lab who's sort of taken on that project. And I look forward to seeing what she's going to find in that system. I'm really excited about these experiments I alluded to in collaboration with Beatrice Hahn's lab, trying to better understand the dynamics of interferon resistance, so how that's changing during these transmission events. Emily Shea is a graduate student here in the lab. She's been adapting the screen to use in a system of HIV latency. So through some really sort of subtle tweaks to the system, we can actually use this same approach to ask about genes that are required to either establish or maintain latency. And then finally, Vanessa Montoya, she's been piloting the screens to try to use whole genome screens to understand comprehensively what the virus uses to replicate inside of cells. And so to just give kind of like a little teaser of what that data looks like, these genes here that are these guides that are scoring on the bottom of these screens. When we look at the gene scores, as an example from our ISG screen shown here, you can see some of the most important sort of required host factors that we know of, and we also can find sort of new genes. So for example, the co-receptor CXCR4 is here, but we also see this gene SEC62. And what's nice is that when we knock out SEC62, we can actually see a really robust decrease of virus infection, suggesting that it is a required gene for HIV. So Vanessa's been kind of piloting using screens to try to understand what the full sort of array of required host factors is for HIV. Okay, and with that, I'm going to end to say thanks to a number of people, including Joy Twentyman and Abby Felton. These are two people here in the lab that work closely with me on these projects. And I refer to us as sort of a subclade of the Emmerman lab, but I will be leaving here soon next summer to start my own lab. And so excited for the new adventures ahead. I have my Twitter handle here, so you can use that to stay tuned if you're interested to find out about what's going on with OhAinle lab as it moves forward. I want to say a big special shout out to Michael Emmerman, who advised me and was my mentor as a PhD student and has allowed me to come back to his lab as a staff scientist now to pursue these projects, and has been tremendously generous. Various folks in the Emmerman lab, as well as other folks at Fred Hutch, some collaborators. I'd be remiss if I didn't point out that I was funded partially in piloting this project through a CFAR grant here at the University of Washington, which was really important for getting this work off the ground. I'm currently funded by an R01 and have joined forces with one of these structural biology centers for HIV, the Cheetah Center. And then last but not least, I always like to quickly mention that I received a supplemental award from the NIH, a re-entry supplement that allowed me to recover from having a lot of kids that were really little, and I actually left science for a while. And this supplemental award actually allowed me to get back into research and to reestablish my career. And so I just wanted to highlight that. And so with that, this is us pre-COVID when we got to do fun things like go on lab hikes together. So this is us at a lake out not far from Seattle. And with that, I will stop and take questions.
I'm going to clap in real life.
Thanks, John.
That was a
So,
brilliantly presented talk and brilliant work. So I'm going to open it up to questions. Fantastic.
hi, this is Doug Richmond. That was really lovely work, and it was very clearly presented. So my question is, I've been following Beatrice Hahn's work and that you described in terms of the longitudinal impact change in the phenotype of interferon sensitivity in an individual
Yep.
with acute infection and then chronic infection and then reactivation. And my understanding from her is that she has not been able to find genetic determinants that account for the phenotypic differences, which is not possible, of course. And so my question is, is your elegant approach able to help dissect this problem? I mean, one difference between your in vitro work and the longitudinal human samples is that any genotypic change has to maintain fitness.
Right.
While a replication, impairment of replication in vitro is, not going to kill a person. So what can you tell about the determinants of this interferon resistance and sensitivity?
Right. Yeah. So this is where this sort of collaboration grew out of with Beatrice, is because she doesn't currently have a good way of trying to figure out what genes are, inhibiting one virus or another. You're right in that the genetics behind it have been complicated. So it's not that she has a panel of viruses where all of them share in common a single mutation in one gene that points in the right direction. And so her data actually suggests that there are multiple things that are going on. And so a virus can become resistant to interferon in one particular way, but it might become sensitive in various different ways. So it might not be that simple. And so what we're hoping is that if we can get the screens working well enough with her viruses, that she'll be able to maybe tease that apart. So she should be able to differentially, like those comparison screens, say that she can pull out what is restricting the different viruses with the different mutations. So we hope to do that soon. I've been working with some of the graduate students and postdocs in her lab, trying to get the screen working in their hands.
Molly, I have kind of a comment question, if I could.
Sure. Favorite type, John. The comment question.
All right. So it's kind of implicit in the screen, right? That to find a restriction factor in this approach, either the virus doesn't have any antagonist or it has a somewhat poorly active antagonist. If it has a poorly active antagonist, the factor can't show up, right? That's really what you see in the Q23. It has such a good VPU that BST2 tetherin's not going to show up.
Exactly.
So can you look for this more directly? Maybe my question would be, could you use viruses that miss certain non-essential accessory genes and see if you then suddenly bring out things that were being antagonized too well for you to pick them up when you use wild type virus?
That experiment has been planned, John, and hopefully will happen soon, because you're right. You would imagine that if you take away an antagonist, so I can say that I've done it with VPU and tetherin. And so if you take away VPU, tetherin jumps way up on the list, even for LAI, suggesting that the LAI VPU has some ability to antagonize its effects. But what we wanted to know is whether other things inhibited by VPU. Tetherin was so high up on the list that we then have now knocked tetherin out of those cells to ask of whatever's left, are there still restrictions present? And so that's an experiment we'd like to do. So you're absolutely right. Any way that we can kind of make a poorly adapted virus by taking away an accessory protein, by making a mutant, by treating with a drug, by using, for example, those SIV strains is something we're trying to get working. So those are not well adapted to human cells. Any of those poorly adapted strains allows us to kind of see what wild type HIV has actually become somewhat adapted to. So you're absolutely right.
I'm just going to chip in one other thing here that we have in the chat. We've got congratulations on the new lab and I put in Berkeley to make sure where you're going, and we'll talk, and And Rick's sister Gail says, thank you. I was even able to follow most of it, enough to know Rick would be so excited about what you and others are doing. Thank you for presenting, and to John for keeping this lecture series going. So I think it was great. It was really nice that you incorporated some of his data too. That was sweet.
I was pretty excited to be able to put that in there. Yeah, that's great. Yeah, thanks, Gail. The fact that you were able to follow a good chunk of it is a high compliment. I'll take that. That's great.
So any other questions? I guess one thing comes to mind is like, this is of course very HIV specific because of the use of lentiviral vector, but have you thought about, is there any way to extend this to other viruses? You'd have to make some different kind of viral vectors and all that, of course, but.
Yeah, as you say, John, it's easiest to imagine porting this over to another like retroviral system where we already have retroviral vectors that can express things and we know how to package them. So that's a fun one to think about. I would love to do it with like acutely infecting viruses. So SARS-CoV-2 comes to mind, or other things. And the answer is it's not quite so easy to do, but I've got some ideas on maybe packaging guide RNAs into those viruses that might work. So those are the kinds of things for sure we'll be trying out in the OhAinle lab in the future.
We are definitely looking forward to those experiments. I mean, it was a beautifully done work, and I'm sure there's more to come. So who else? Anyone else have any thoughts? Questions?
Thanks to everyone for coming.
Yeah, thank you for coming.
Yeah, happy to be with you. Yeah.
Very good. Well, thanks again. It was terrific. Really.
All right, cheers.
Mary Lewinsky, and great to see you. All right. Take care then, everybody.
Thanks, everyone.
Bye-bye now.
Bye.